Welcome, everyone. I'm excited to kick off our presentation today. The topic is nucleotides and nucleic acids, and we'll be exploring the fundamental building blocks of life. On this first slide, we see that a gene is a segment of a DNA molecule that contains the information needed to produce a functional biological product, whether that's a protein or RNA. Typically, a cell has thousands of these genes, and since DNA molecules are quite large, they can store a vast amount of information. Interestingly, the primary functions of DNA are to store and transmit biological information. So, over the course of this presentation, we'll delve into the structure of nucleotides and nucleic acids and understand how they underpin genetics and molecular biology. Nucleotides have three main characteristic components that define their structure. First, there's a nitrogenous base, which contains nitrogen atoms and is essential for the genetic code. Second, there's a pentose sugar, which provides the backbone for the nucleotide. And third, there's a phosphate group that links the nucleotides together in the chain. When you look at the molecule without the phosphate group, it's called a nucleoside. Now let's talk about these nitrogenous bases. They are derivatives of two main parent compounds, pyrimidine and purine. Both DNA and RNA contain two major purine bases, which are adenine, abbreviated as A, and guanine, G. In addition to purines, both molecules also have pyrimidines, but there's a key difference. In both DNA and RNA, one of the pyrimidines is cytosine, or C. However, the second pyrimidine isn't the same in both molecules. In DNA, it's thymine, T, while in RNA, it's uracil, U. This difference is one of the fundamental distinctions between DNA and RNA. Nucleic acids also contain two types of pentose sugars. DNA's recurring units are based on 2-deoxy-D-ribose, which means it's missing an oxygen atom at the 2 position. RNA, on the other hand, contains D-ribose, which has that oxygen. So, to sum up, nucleotides are the building blocks of nucleic acids, and their structure is defined by these three components: the nitrogenous base, the pentose sugar, and the phosphate group. The variations in bases and sugars are what give DNA and RNA their unique properties and functions. RNAs have a much broader range of functions than you might initially think, and in fact, several different classes of RNA are found within cells. For example, ribosomal RNAs, or rRNAs, are essential components of ribosomes, which are the molecular machines responsible for protein synthesis. Without rRNAs, ribosomes couldn't function properly. Then we have messenger RNAs, or mRNAs, which act as intermediaries. They carry genetic information from the DNA, typically from one or a few specific genes, to the ribosome. This is where the actual protein synthesis takes place, so mRNAs are crucial for translating genetic instructions into functional proteins. Finally, transfer RNAs, or tRNAs, are like the adapters of the process. They faithfully translate the information encoded in the mRNA into a specific sequence of amino acids, which are the building blocks of proteins. Overall, these different types of RNA work together in a coordinated way to ensure the accurate production of proteins, highlighting how versatile and vital RNA molecules truly are in cellular function. Today, we're going to focus on nitrogenous bases, which are essential components of DNA and RNA. These bases are the building blocks of genetic information. They come in two main categories: purines and pyrimidines. Purines include adenine and guanine, which are larger molecules with a double-ring structure. Pyrimidines, on the other hand, include cytosine, thymine, and uracil, which have a single-ring structure. The specific pairing between these bases, adenine with thymine or uracil, and cytosine with guanine, is crucial for the stability and replication of genetic material. Understanding these bases helps us comprehend how genetic information is stored, copied, and transmitted across generations. They are the key to the molecular code that makes life possible. Let's take a closer look at nucleotides, which are the fundamental building blocks of DNA and RNA. These molecules are essential because they carry the genetic information that defines living organisms. Each nucleotide is made up of three main components: a sugar molecule, a phosphate group, and a nitrogenous base. The sequence of these bases encodes the instructions necessary for life. The key point to remember is that the order of these bases, adenine, thymine, cytosine, guanine in DNA, determines genetic information. This sequence is what allows cells to produce proteins and carry out their functions. Understanding nucleotides helps us grasp how genetic information is stored, replicated, and expressed. It's the foundation of molecular biology and critical to many applications, from medical research to biotechnology. So, in the context of our overall presentation, recognizing the structure and function of nucleotides is crucial because it connects to how genetic information is inherited and how it can be manipulated for various scientific advancements. Let's take a closer look at the goals for this section. Our main aim is to understand the basic mechanisms behind transcription, RNA processing, translation, and replication. These are fundamental processes that drive how genetic information is expressed and maintained in cells. Now, moving on to the topics we'll cover. First, we'll explore the structure of nucleic acids, which form the backbone of genetic information. Next, we'll dive into how protein-coding genes are transcribed into mRNA and how this mRNA becomes functional. We'll then examine how tRNAs decode this mRNA during protein synthesis, translating the genetic code into amino acid sequences. Following that, we'll look at the step-by-step process of protein synthesis on ribosomes, which is essentially how proteins are built in the cell. Afterwards, we'll discuss DNA replication, the process by which DNA copies itself to ensure genetic information is passed on accurately. Finally, we'll touch on basic molecular genetic mechanisms, especially focusing on Part A of this topic, to give you a comprehensive understanding of how these processes are interconnected. Throughout this presentation, I want you to keep in mind that these mechanisms form the core of molecular biology. They explain how genetic information flows from DNA to functional proteins, maintaining life at the cellular level. Nucleic acids are polymers, which means they are long chains made up of smaller units called nucleotides. As you can see in Figure 4.2a, the backbone of these molecules is composed of repeating units of phosphate groups linked to deoxyribose or ribose sugars. These sugar-phosphate units form the structural framework of the nucleic acid chain. The nucleotides are connected to each other through phosphodiester linkages, which are essentially covalent bonds that form between the phosphate group of one nucleotide and the sugar of the next. This connection is crucial because it holds the chain together in a stable, linear fashion. Just like proteins, nucleic acid chains have a directionality, which we describe as having a 5-end and a 3-end. This polarity is important because it influences how the strands are read and replicated. By convention, when we write the sequence of bases in a strand, we always do so from the 5-end on the left to the 3-end on the right. This is shown in Figure 4.2b. Recognizing this directionality helps us understand how genetic information is stored and transmitted, which ties back to the overall theme of how biological molecules carry and pass on genetic information effectively. Now, in the most common conformation of the double helix, known as bDNA, you'll notice that approximately 10 base pairs occur per turn of the helix. This regular pattern is fundamental to how the structure maintains its stability and flexibility. Within this structure, there are two distinct grooves, a wide, major groove and a narrower, minor groove. These grooves are crucial because they expose parts of the bases to the outside environment, making them accessible for interactions. This is where DNA binding proteins come into play. They locate and interact with specific sequences of bases that are exposed in these grooves, which is essential for processes like gene regulation. Moving on, cellular DNA predominantly exists as a right-handed double helix, which you can see in Figure 4.3. The two strands are interwound and anti-parallel, meaning they run in opposite directions. These strands are complementary, pairing specifically through Watson-Crick base pairs, adenine with thymine, and guanine with cytosine. It's also important to note that the sugar-phosphate backbones of these strands are on the outside of the helix, providing structural support. Meanwhile, the bases are stacked inside, creating a stable, stacked arrangement that's key to the molecule's integrity. Understanding this double helical structure is fundamental because it underpins how genetic information is stored, accessed, and manipulated within the cell. Watson and Crick proposed that native DNA consists of two anti-parallel chains arranged in a right-handed double helix. This means that one strand runs in one direction while the other runs in the opposite direction, creating a stable and specific structure. Inside the helix, complementary base pairs, adenine pairing with thymine and guanine pairing with cytosine, are formed through hydrogen bonds. These base pairs are stacked perpendicular to the long axis of the helix, spaced about 3.4 angstroms apart, and there are roughly 10.5 base pairs per turn of the helix. It's also important to recognize that DNA isn't limited to just one structural form. Besides the classic bDNA, which is the most common form, there are variations like aDNA and zDNA, each with slight differences in shape and conformation. Some sequences can cause the DNA to bend or form unusual structures, which can be important for how DNA functions in the cell. Additionally, specific sequences allow DNA to fold into structures such as hairpins, cruciforms, or even triplexes and tetraplexes. These structures are not just curiosities. They can play roles in regulation, replication, and stability of the genetic material. Overall, the structure of double helical DNA is fundamental because it underpins how genetic information is stored, replicated, and expressed in all living organisms. DNA can be bent because it lacks stabilizing bonds oriented parallel to the axis of the double helix. This means that the structure isn't rigid and can flex, which is quite important for its function. Bending often occurs when transcription factors, like the TATA box-binding protein, bind to specific DNA regions. For example, as shown in Figure 4.5, these proteins induce bending to facilitate the process of gene expression. Similarly, DNA-binding proteins such as histones also cause DNA to bend, which helps in organizing and compacting the genetic material. This bending is absolutely crucial for packaging DNA into chromatin inside eukaryotic cells. Without this flexibility, it would be much more difficult to fit all that DNA into the nucleus efficiently. So, in essence, DNA's ability to bend plays a key role in gene regulation and chromatin structure, tying back to our overall theme of how DNA's physical properties influence its biological functions. In many cases, multimeric proteins can reach incredibly large sizes, sometimes involving tens or even hundreds of subunits. These massive complexes are a perfect example of what we call supramolecular structures, which represent the highest level of structural organization in biology. Think of complexes like the mRNA transcription pre-initiation complexes, ribosomes, proteasomes, and spliceosomes. What's fascinating is that these supramolecular complexes often function as what we call macromolecular machines. This means that the individual subunits don't work in isolation. Instead, their activities are coordinated in a way that allows the entire complex to perform a specific, overall task. For instance, in protein synthesis, the ribosome acts as a machine where all the parts work together seamlessly to produce proteins efficiently. So, these structures highlight how nature has evolved incredibly intricate and organized systems to carry out essential biological processes at a very high level of complexity. Let's delve into how DNA and RNA differ, particularly in the sugars they contain within their nucleotide monomers. DNA has deoxyribose, which lacks a hydroxyl group at the 2 position, while RNA contains ribose, which has that 2-hydroxyl group. This small difference has significant implications for their stability. The phosphodiester bonds in RNA are more vulnerable to hydrolysis, especially in basic solutions, because the 2-hydroxyl group can act as a nucleophile. This allows it to attack the phosphate backbone, leading to bond cleavage, a process known as base-catalyzed hydrolysis. In contrast, DNA's phosphodiester bonds are much more resistant to this kind of attack because it doesn't have the 2O group. This makes DNA a more stable molecule over the long term, which is probably why it was selected as the primary molecule for storing genetic information. So, in essence, the presence of the 2-hydroxyl in RNA makes it more reactive and less stable, whereas DNA's lack of this group contributes to its durability for preserving genetic data across generations. Let's talk about DNA denaturation. This process involves the unwinding and separation, or melting, of the 2 strands of the double helix. In living cells, enzymes like helicases are responsible for doing this job. But in the lab, we can induce denaturation by heating the DNA, as shown in Fig. 4.7, or by using buffers with low ionic strength or extreme pH levels. One interesting way to monitor this process is through UV absorption spectroscopy, which increases as the DNA strands separate. Now, the stability of the DNA strands depends on their G+C content because GC base pairs are held together by 3 hydrogen bonds, making them more stable, while AT pairs have only 2 hydrogen bonds. That's why the melting point of DNA increases with higher G+C content, as illustrated in Fig. 4.7b. Finally, when conditions are reversed, cooled or neutralized, single strands can come back together and reform the double helix in a process called renaturation, or annealing. This ability to denature and then reanneal DNA is fundamental to many molecular biology techniques and helps us understand DNA's dynamic nature. Nick, processes such as replication that unwind double helical DNA introduce torsional stress, which results in supercoiling. This phenomenon is most evident in circular DNA molecules, like bacterial plasmids and some viruses, but it also occurs in linear eukaryotic chromosomes. When DNA is being replicated, it causes overwinding ahead of the strand separation site, leading to positive supercoiling. Conversely, underwinding can happen, resulting in negative supercoiling. To manage this torsional stress, cells use enzymes called topoisomerases. Topoisomerase I relaxes supercoiled DNA by cutting one strand and allowing it to unwind. On the other hand, topoisomerase II can introduce negative supercoils into DNA, which actually helps in relieving supercoiling tension. In fact, cellular DNA naturally tends to be slightly negatively supercoiled. This negative supercoiling is important because it facilitates vital processes like replication and transcription, where the DNA needs to be unwound easily. Overall, supercoiling and the enzymes that regulate it are crucial for maintaining DNA stability and function during cell activities. Most cellular RNAs are typically single strands, but what's really interesting is that they often form double helical regions when complementary sequences come together. These secondary structures are quite common and play crucial roles in RNA function. 2 of the most well-known types are hairpins and stem loop structures, which you can see in Fig. 4.9. In these structures, the stem regions actually form double helices similar to those in DNA, specifically aDNA. The turns that connect these helices are shorter in hairpins compared to stem loops, which influences their overall shape and stability. Now, beyond these simpler structures, RNA molecules can fold into very complex 3-dimensional shapes. For example, tRNA and rRNA are known for their elaborate 3D architectures, which are essential for their functions in protein synthesis. One fascinating example is the pseudoknot structure, like the one seen in human telomerase RNA, shown in Fig. 4.9b. These pseudoknots add an extra layer of complexity and stability to the RNA's architecture. And speaking of complexity, the 3D structures of ribozymes, these RNA molecules that act as enzymes, are incredibly intricate. In fact, they can rival proteins in their structural sophistication. This highlights just how versatile and sophisticated RNA molecules are, not just as carriers of genetic information, but also as functional, catalytic entities within the cell. Messenger RNA is just one type of several classes of cellular RNA. Transfer RNAs, or tRNAs, play a crucial role as adapter molecules during protein synthesis. They are covalently linked to an amino acid at one end, and they pair with mRNA in such a way that amino acids are added to a growing polypeptide chain in the correct sequence. Ribosomal RNAs, or rRNAs, are the structural and functional components of ribosomes, which are the sites of protein synthesis. There are also many other specialized RNAs with different functions. For example, some RNAs called ribozymes actually have enzymatic activity, meaning they can catalyze chemical reactions themselves. Now, in prokaryotic cells, a single mRNA molecule can sometimes code for multiple polypeptides. If it encodes just one, we call it monocistronic, which is typical for eukaryotic cells. But if it encodes two or more different polypeptides, it's called polycistronic. In contrast, most eukaryotic mRNAs are monocistronic, meaning each mRNA usually codes for just one gene or one polypeptide. And just to clarify, when I say cistron, I'm referring to a gene in this context. So, understanding these different types of RNA helps us appreciate how genetic information is expressed and regulated within the cell, which ties back to the overall theme of gene expression and protein synthesis. In transcription, a specific sequence in the DNA is copied into RNA through a process that is catalyzed by an enzyme called RNA polymerase. The RNA that is synthesized is actually complementary to the DNA template strand, meaning it pairs with it in a specific way, and runs in the opposite direction, or anti-parallel, to the template strand. Now, the RNA sequence you get is the same as the coding strand of DNA, which isn't shown here but is an important point. An interesting aspect of this process is that the growth of the RNA chain happens in the 5-3 direction, just like DNA synthesis. The actual chemical reaction involves adding nucleotides to the growing RNA chain, and this process proceeds with the elimination of inorganic pyrophosphate, which drives the reaction forward. It's also important to note that the transcription start site is numbered +1, serving as a reference point. Sequences before this site are called upstream and are located before the start, while sequences after the start site are downstream, positioned further along the gene. Understanding this mechanism is fundamental because it connects to how genes are expressed and how genetic information is transferred from DNA to RNA, which ultimately guides protein synthesis. The primary RNA transcript produced during transcription has the same sequence as the coding or non-template strand of DNA. This means that if you look at the DNA, the RNA sequence essentially mirrors the coding strand, with the exception of uracil replacing thymine. It's important to note that this transcript is actually complementary to the template strand of DNA, which is the strand that RNA polymerase reads to synthesize the RNA. Now, the promoter is a key DNA sequence located in the non-template strand. Think of it as a molecular signpost that indicates where transcription should begin. It tells RNA polymerase exactly where to start, marking the +1 site, which is the first nucleotide to be transcribed. Understanding these conventions helps us grasp how genes are transcribed accurately, ensuring the correct RNA is produced to ultimately make the right proteins. It all ties into the bigger picture of gene expression regulation and how cells control which proteins are made and when. Now, let's take a closer look at how transcription by RNA polymerase unfolds in 3 main stages: initiation, elongation, and termination. On this slide, we're focusing on the initiation stage, which is particularly important because it's where the regulation mostly happens. During initiation, RNA polymerase binds to a specific region of DNA called the promoter, and it often needs some help from transcription factors to do this effectively. Once bound, about 14 base pairs of DNA are melted, creating what's known as the transcription bubble. This is a critical step because it exposes the template strand, allowing the enzyme to start synthesizing RNA. At this point, RNA polymerase begins to assemble a short RNA chain, just 2 nucleotides long, that is complementary to the DNA template strand. Understanding this initial step is key because it sets the stage for the entire process of gene expression. After initiation, RNA polymerase moves forward and clears the promoter region, leaving behind the transcription factors that helped start the process. As it progresses, it maintains a bubble of about 14 nucleotides where the DNA strands are separated, allowing the enzyme to read the template strand and synthesize the RNA. The RNA grows in the 5 to 3 direction, which is the direction of synthesis. The new RNA strand stays hydrogen bonded to the DNA template strand through its last 8 nucleotides, forming a stable hybrid region. In eukaryotic cells, this elongation process is quite rapid, occurring at approximately 1,000 nucleotides per minute. This efficient process is essential for quick gene expression and proper cellular function. This slide doesn't contain enough information. That's why we're moving to the next slide. When RNA polymerase reaches a termination sequence, it signals the end of transcription. At this point, transcription stops, and the polymerase along with the newly formed primary transcript are released from the DNA. This release is a crucial step because it allows the RNA polymerase to be recycled and initiate transcription again at a different promoter site. Essentially, once the process is complete, the polymerase is free to start the whole process anew, enabling the cell to produce multiple RNA molecules from the same or different genes. This termination process is vital for regulating gene expression and ensuring that transcription occurs efficiently and accurately. So, about half of the genes in prokaryotic cells are organized into what we call transcription units, or operons. Now, an operon is a set of genes that are transcribed together from a single promoter, which means they share a common regulatory region. This setup usually includes genes that work together in a particular process, like building the amino acid tryptophan. For example, in E. coli, there's the TRP operon, which is a classic example. The interesting thing about the TRP operon mRNA is that it's polycistronic, meaning it contains multiple coding sequences, specifically 5 different genes. Each of these genes, called cistrons, is translated into its own protein. So, in essence, a single mRNA molecule can produce several proteins that function together, which is quite efficient. This organization is a fundamental feature of prokaryotic gene regulation because it allows bacteria to quickly respond to environmental changes by turning on or off entire pathways at once. Connecting this to the broader theme, understanding operons helps us see how bacteria coordinate gene expression efficiently, which is quite different from how eukaryotes typically organize their genes. Most eukaryotic genes are transcribed separately, even in simple organisms like yeast. For example, the TRP biosynthesis genes each have their own promoter, which means they are individually regulated. Interestingly, these genes are encoded on different chromosomes in yeast, highlighting the complex organization of eukaryotic genomes. Additionally, in higher eukaryotes, gene coding sequences are often interrupted by non-coding regions called introns. These introns are not part of the final protein product. Before the mRNA is functional, these introns are removed through RNA processing reactions, ensuring that only the necessary coding sequences are translated into protein. This organization allows for greater regulation and complexity in gene expression, which is a key theme in understanding how eukaryotic cells control their genetic information. Now, let's talk about what happens to eukaryotic pre-mRNAs before they become fully functional. Unlike bacterial mRNAs, which are ready to go as soon as they're transcribed, eukaryotic pre-mRNAs need a lot of processing. One of the first and most critical steps is called capping. This involves adding a special structure called the 5' cap to the beginning of the pre-mRNA. This cap is known as the 7-methylguanylate, or M7GPPP. Essentially, a methylated guanine nucleotide is attached to the first nucleotide of the pre-mRNA via a unique 5' linkage. This isn't just a decorative add-on. It plays several vital roles. For instance, it helps the mRNA get transported from the nucleus to the cytoplasm, which is crucial for protein synthesis. Additionally, the 5' cap protects the mRNA from being degraded by nucleases, which are enzymes that break down nucleic acids. Without this protection, the mRNA wouldn't last long enough to be translated into protein. Finally, the cap is important for initiating translation. It acts as a recognition signal for the ribosome, helping it to identify and bind to the mRNA efficiently. So, in summary, this capping process is a key modification that ensures the mRNA is stable, properly localized, and ready for translation, all essential for proper gene expression. Now, let's take a look at how eukaryotic pre-mRNAs are processed after they are transcribed. First, at the 5' end, the pre-mRNA is capped, which is crucial for stability and for the initiation of translation. Simultaneously, at the 3' end, the pre-mRNA undergoes a modification called polyadenylation. This process involves cleaving the longer pre-mRNA at a specific site called the polyadenylation site. After that, up to 250 adenylate residues are added by an enzyme known as A polymerase, which extends the tail in a template-independent manner. Along with these modifications, non-coding intron sequences are excised through splicing, and the coding exon sequences are ligated together to form the mature, functional mRNA. This mature mRNA retains untranslated regions, or UTRs, at both the 5 and 3 ends, which play roles in regulation. The A tail, in particular, is important because it helps protect the mRNA from degradation by nucleases, thus ensuring the stability of the message as it makes its way out of the nucleus for translation. Overall, these modifications, capping, splicing, and polyadenylation, are essential for producing a stable and translatable mRNA, which ties directly into the broader theme of gene expression regulation. Let's delve into how protein domains are encoded within genes. Often, a single exon or a small collection of exons within a larger gene can encode a specific protein domain. This means that the building blocks of proteins are modular and can be mixed and matched in different ways. Now, what's fascinating is that these coding regions for domains can be included or excluded from the final messenger RNA through a process called alternative splicing. This process essentially allows a single gene to produce multiple different mRNA molecules, each coding for a different protein variant, known as isoforms. This diversity is very important because it provides a way for cells to regulate gene expression in different tissues or under different physiological conditions. For example, it's estimated that about 60% of all human genes are expressed as alternatively spliced mRNAs. To put this into context, let's look at the fibronectin gene, which is a great example illustrated in Figure 4.16. Depending on how the exons are spliced, you get different isoforms of fibronectin. Specifically, the fibroblast and hepatocyte isoforms differ in their content of the EIIIA and EIIIB domains, which are crucial for cell surface binding. In fact, researchers have identified around 20 different isoforms of fibronectin that result from alternative splicing. This highlights how versatile and dynamic gene regulation can be, allowing a single gene to fulfill multiple roles depending on the tissue type or physiological state. So, overall, alternative splicing is a key mechanism that greatly expands the functional diversity of our proteome and plays a vital role in gene regulation across different contexts.